So Adrienne is a member of my lab for now. She's going to be changing into Paul's domain soon. She got her PhD in marine sciences from the University of North Carolina in Chapel Hill. She's currently working on building a deep machine learning pipeline to learn the language to describe the metagenomic world, hopefully in terms of its functionality, including metal binding and enzymatic or oxidoreductase activity.
All right, thank you, Anna. So today I'm going to be talking about my work trying to apply deep learning to the relationship between microbial communities and their environment. I've been interested for a long time in how microbes interact with their environment to regulate biogeochemical cycles and how we can capture this very, very complex relationship better in our models, in particular in settings where we have very poor annotation of the genes and the genomes that are involved. And I am particularly interested in applying deep learning and a concept called transfer learning to this. So I'll be talking about that effort today. The basic idea is, even if we don't know what a particular sequence does, what exactly defines the functional or biogeochemical relevance of a random sequence you might grab out of the environment is the context of all the sequence around it. So that's true for the individual sequencing read, what other nucleotides are on that particular sequence, and also in the setting of an environment in a community, the other sequences from all the other genomes, that context matters as well. So you can, even without knowing anything about what a sequence does or where it comes from, you can sort of formalize this idea by reframing the question of, okay, well, if I have a sequence and I block out a section of that sequence, can I, just based off of the surrounding context of the sequence alone, predict what sequence should be there, what nucleotide should be there? And as I scale this up, and assuming you come up with a functionally and evolutionarily relevant representation of that sequence based off of that sort of that modeling task, can you then scale this up to predict functional targets of interest at the level of a sequencing read or of a protein? Or as you combine this to start thinking about microbial communities, to start being able to model these sort of complex community phenotypes like biogeochemical outcomes, or the relationship between the environmental geochemical context of the community with the community that's living there. So there were sort of three major objectives to this postdoctoral fellowship that I started last year, and I'm continuing through the next year or two. The first was to do this general task of, can I learn the complex functionally and evolutionarily relevant features that define biological diversity as a whole from all the sequences that we have, regardless of where they come from. And that's where the deep learning comes in. And then using a concept called transfer learning to leverage these deeply learned features that describe biology as a whole to predict more specific tasks of interest where we might have a lot less data with nicely curated labels. So at the read or protein level, you're thinking about, can I predict novel functional targets of interest, for example, or motifs that we're interested in? And as we scale that up to a community level, can you predict the geochemical conditions associated with environmental metagenomes that you sample? So starting to map that relationship between a microbial community and their environmental context. And finally, once you have these models, can you use them to predict or validate known protein motifs that characterize either a function of interest or the geochemical context of a microbial community, whether that's in sort of modern Earth conditions and environments that we see now, but you can also imagine doing this, if I have a putative geochemistry at a point early in Earth's history or even on another planet, can I make some predictions about what sorts of functional motifs I expect to see there? So this is a pretty ambitious 3-year project. So today I'm going to be talking about more, the first half of that, which is the general model of trying to capture these features that define biological diversity as a whole, and then at the read level, trying to apply this to identify novel functional targets of interest using transfer learning. So just to go over real quickly what transfer learning is, when you think of a modeling task, usually you say, okay, I have a data set and I want to predict something associated with that data set. And that's what I'm showing here on the left. What's very difficult, especially in environmental settings, is that it can be very hard to get really good labels of the thing that you're interested in. It's very time intensive and expensive to functionally characterize genes. So a lot of genes remain unannotated. It's very hard to go out into the environment and gather samples, get those labels. So often it can be really hard to learn these super complex functions that define environmental systems. So my idea was to use transfer learning. So the basic concept is that if you have a giant data set, so say all the sequences that we have that have been sampled, regardless of where they're coming from, if you can frame that such that you can model some learning task, maybe it's not the learning task you're necessarily interested in, but it's some learning task that you can do with that giant data set, you can learn some of these general features that define biological systems in general. Once you have that domain knowledge, you can just use that as a starting point for your modeling task. So now you have your smaller data set. You start with those features that define the general biological features, and then you are able to learn faster and more accurately your learning task of interest, where before you may not have had enough data to get a good model out of it. And the reason that this works is that deep learning models in general learn complex functions successively from universal to more task-specific features. So I'm showing you an example from the computer vision world here, but this applies to all domains. There is a long-established modeling task in computer vision, taking a data set called ImageNet, which is this massive data set of labeled images, these painstakingly manually labeled images of I think 1,000 image labels and millions and millions of images. And the state-of-the-art model that can predict the image classification based off of these images is, I'm showing a portion of it here. And what it does, it's actually a 12-layer model that are all stacked together. The earliest layers learn really general features associated with images. Like I'm showing here on layer 1 on the far left side, it's really just an edge detector. It sees edges. In layer 2, you start to kind of put these together in more complex ways. You're seeing corners, stripes. Layer 3, you start to see more complex geometries. And only as you get to layer 4, you're starting to see like really task-specific labels, like it's detecting dog faces or bird legs, something that's really specific. And this is actually a 12-layer model. So successive layers are detecting different breeds of dogs or different kinds of birds, that sort of thing. So you can imagine if you are somebody coming from the medical imaging world and you want to be able to say, is this image of a cell cancerous or not, or something like this. It's really expensive and difficult to get images of these cells, and you don't have a lot of labels. But you don't necessarily need to train from scratch, like this is what a corner is, this is what a curve is. You can use this pre-trained model from ImageNet as a starting point, and then you can get to a much better model of the thing that you're actually interested in much faster, and potentially making it feasible where it would have been infeasible before. So I'm working on applying this idea to biological systems, and we're really missing both sides of this equation, both this general model, what I call a universal language of life that can use potentially a huge diversity of biological sequences that are available in public databases. It turns out you don't actually need all of the sequences that are there. If you choose your data set really carefully, you can use a much condensed data set, but a much greater diversity than you would ever otherwise have if you were relying on having well-labeled data sets. And learn a general model that I'm calling Looking Glass, where it's this task where you take a sequence, you mask out a nucleotide, can you predict what nucleotide should be there? And from that, you're learning just general functionally and evolutionarily relevant features of sequences. And you take that domain knowledge, and then you can do the other side of this equation where you fine tune this original Looking Glass model. Say you have a sequence with a particular function of interest that you're trying to label. You can then fine tune that Looking Glass model to classify, is this my function or not? Something like that. So that's exactly what I did. I went ahead and I took a broad swath of the biological diversity that are available in public databases, chopped it up into sort of read length chunks, the kind of sequence that you get out of the environment, these sort of 150-ish base pair chunks, and trained a language model to recognize the masked nucleotide. And the hope, at least, is that you're going to produce a model that captures these functionally and evolutionarily relevant features that underlie biology as a whole. So what I want to show in the next couple slides is the fact that it does that. So this slide is showing the functional relevance of this model. So I took Looking Glass and I took some sequences that had functional annotations based off of my phaser annotations. And I split them up so that there was as little overlapping sequence identity as I could. And I trained it with just few epochs to predict the functional annotation of that read. And it was able to do that to the 4th EC number with 77% accuracy. What I'm showing here on the left is the Sankey diagram between the true predictions on the left side and the predicted annotations on the right side. And the number is the 1st EC number. So it's able, the 4th EC number, it's getting 77% accuracy. For the 1st EC number, it's getting much higher than that, so a little over 90, I believe. And so that tells us that it's able, with very little effort, to capture that functional relevance of a sequence. And even at a sort of higher order functional feature, you can map from that EC number to the metal cofactor identity of that sequence. And you're able to get that at about 76% accuracy. And this is not specifically predicting metal binding. It's just based off of the functional annotation, I'll note, but it just indicates that there's higher order functional relevance of the embeddings of these sequences, of these feature references.
So what was the input features in this case in your model?
It's just the raw sequence.
So actual sequence. Were you
DNA sequence?
Did you have to trim the length of the sequence so that it has the same vector?
You use something called, these are LSTMs. If you're a deep learning person,
yeah, okay.
You basically use backpropagation through time. So it ended up, I was using 100 base pair length.
Okay, so it's a convolutional neural network coupled with LSTM, right?
It's just 3-layer LSTM.
Oh, okay. All right.
Yeah, so no CNN part. But,
all right.
Yeah. So that's the functional relevance of those sequences. But I also wanted to be able to capture evolutionary relevance of sequences. So what's really cool about these embeddings, all I did is I took homologous pairs of sequences and non-homologous pairs of sequences. I plugged them into the original Looking Glass model. I did 0 fine tuning, and I just compared the output embedding. You take a sequence and it outputs a sort of fixed vector, what I call embedding. In this case, it's a 104-place embedding. And you can compare how similar in these embeddings, just like you would with a raw DNA sequence. You can say how similar are these sequences. You can do the same, just compute the cosine similarity between 2 embeddings and how similar are they. And between homologous pairs of sequences, there's much higher embedding similarity than there are between non-homologous sequences. And this is, again, with 0 fine tuning. I'm sure you could get better performance than this if you were to actually specifically fine tune it to this. But you're able to get 80% accuracy if you just pick an embedding similarity threshold to differentiate between homologous or non-homologous sequences. And this is true, I'm showing this at the genus level, but this is true at all phylogenetic levels, at the class level, at the phylum level. And this is especially cool, I think, in the context of embedding similarity is not necessarily correlated with raw sequence similarity. So I'm showing here the embedding similarity versus the just the raw bit score. So just a metric of like the nucleotide overlap of those sequences. And not only are they not well correlated, but there's actually a pattern here. So if there was 0 relationship between sequence similarity and embedding similarity, you'd see sort of a wash of dots all across this graph, but you don't. I've sort of drawn in this dashed line to indicate this. At really high sequence similarity, the embedding similarity is also high. And that makes sense because you're going to, very, very, very similar sequences are probably going to be homologs, and that makes sense. But there are a lot of homologous sequences where the sequence similarity is actually very low. And in these cases, the embedding similarity is often much higher. So we're not just memorizing the order of sequences, we're capturing evolutionarily relevant features of sequences, something about homology that I think is a little bit more interesting and more useful for downstream prediction tasks. So finally, the last thing I wanted to show about the looking glass model is just when you go to different environments, you would hope that the functional differences and the compositional differences of those environments would also be captured. And that's what we see here. So this is, again, I took a bunch of sequences from different environments just based off of the environmental package, which is like a sort of a very broad metadata category of like, is this soil or is this from water? Really, really general category. And if you compare the, and then this is a TCE plot just showing how, trying to separate out how similar sequences are, or embeddings of these sequences are across environments. And so the colors here are indicating the different environmental packages. And this is again with zero fine tuning, you start to see this separation across environments. So the embeddings of these sequences are capturing sort of distinct signatures for different environmental contexts as well. So great, we have a model that can capture these biological features of interest that are common to all of biology, but we want to be able to apply this to something downstream. So what I did in this case was to, I'm just going to show one of these examples, although there are many, many functional examples that you could think of testing. I wanted to see, okay, if I go into an environment, can I identify novel, completely unseen oxidoreductases for which we don't have annotations? So I went into SwissProt, this manually curated database of proteins associated with their functions, and I took out all the oxidoreductases and I split them up so that none of the sequences were more similar by more than 50% amino acid identity. So they're really, really quite different between the training and the validation set. So the sequences, the oxidoreductases that I'm actually training the model on are very, very different looking than the ones in the validation set, but they do share this functionally, the fact that they're all oxidoreductases. And this is particularly relevant for oxidoreductases because they're this very ancient group of enzymes, and they often have very low sequence identity within orthologous groups. And I was able to fine tune this model. So I started with looking glass. I did a fine tuning procedure to predict, is this sequence an oxidoreductase or not? And I was able to predict oxidoreductase identity with 80% accuracy. And this is again with tests, I'm showing the 80% accuracy in the validation set where the oxidoreductases are very different than the ones that we were training on. So it's capturing something about the functional features underlying oxidoreductases. And what's great about this is that, so you have this overall 80% accuracy that you're seeing on the far left of this figure here. But if you were to say, want to test unannotated putative oxidoreductase in the lab, you want to be pretty sure that you're going to have a good hit rate. So you can toggle what you're actually predicting when you say oxidoreductase is not, is something on the scale of 0 to 1. I'm saying anything above 0.5 on the far left here is an oxidoreductase, and that has 80% accuracy. But if you say, okay, anything above a 0.99 probability prediction is an oxidoreductase, I'm going to have, for anything I identify as an oxidoreductase, I'm going to have 99% certainty that that's actually an oxidoreductase. So if you wanted to do some experimental validation of these predictions, you can sort of toggle your probability threshold to meet the needs that you have. So I just wanted to demonstrate how this could be used in the environment. In environmental settings, microbial dark matter, quote unquote, really dominates these settings. So if you go out to, I went and grabbed a metagenome from the TARA project that is from the DCM and the mesopelagic depths in the South Pacific Gyre. So this very sort of resource limited marine environment. And tried to annotate it and see how many EC1s I could get out of it. I'm using MyPhaser here, which is pretty stringent, but it's common to pretty much all functional annotation approaches to get really, really high proportions of just completely unannotated reads. So in this case, in the DCM, I was only able to get 2% of reads that were annotated at all, and about 0.45% of those were were identified as EC1s, as oxidoreductases, and it was even fewer in the mesopelagic, in the deeper depths, which is quite common as well. But if you apply my model, you see that about 15 to 18% in these samples are predicted to be EC1s. And this just sounds reasonable to me. It seems reasonable that of the proteins that are present in an environment, about 15% of them would be oxidoreductases. That sounds about right. And you can, again, I've highlighted in the different shades of green, you can toggle between that expected precision level. So depending on what you want to do to analyze this result, to sort of do your downstream analysis of it, you can choose a different cutoff and look at different subsets of those EC1 proteins. But you can identify 5 to 20 times more oxidoreductase at the 95 to 99% precision level than you otherwise would. And you can get 100x more if you're looking at that overall 80% prediction. So depending on what your downstream purpose is, you can choose different precision levels, but the idea, but there are lots of things that you could potentially do with this. So say you want to compare the oxidoreductase diversity or composition across different environments, you're potentially losing a lot of information when you're just looking at this 0.45%. So maybe by expanding this to 15% of the sequences that you're looking at, you can reduce your bias and look at a much more diverse and complete set of the oxidoreductases that are there. You can maybe consider really high precision predictions to target for functional characterization. So we mentioned earlier Nolan's project trying to map the redox potential of sequences to the evolutionary history of oxidoreductases. The oxidoreductase tree can be very hard to resolve phylogenetically. So perhaps by picking out a greater diversity of oxidoreductase sequences, we can expand the diversity of oxidoreductases we see and more finely resolve this tree. And finally, you can potentially sort of analyze sequences, look at where the model is looking on that sequence to decide what is driving that functional prediction and try to identify important motifs that are driving the predictions of the models. So just to wrap up here, hopefully I convinced you that Looking Glass is capturing these functionally evolutionary relevant features of biology. And there are many potential downstream functional targets. I showed you one with oxidoreductases today, but I'd love to talk to people about their own applications and whether they have something they want to look at. And finally, we can also sort of analyze some of these predictions to identify sequence motifs that are driving functional predictions, and hopefully identify something that's something that we haven't captured with other approaches. So for the next year or so, I'll really be thinking about scaling this up to the community level, linking geochemical conditions associated with environmental metagenomes, and that's going to be a really big, both a difficult scientific problem, but also a big computational challenge to take all those reads and figure out how to represent a community in a useful way. But there's a lot of things that we can think about doing with this, generating sequences that are characterizing particular geochemistries, and starting to model some of these more complex community level phenotypes, like rates of biogeochemical processes, for example. So with that, I will wrap up. Thank you so much to the ENIGMA team and the Bromberg Lab. It's been a very inspiring year, and I'm looking forward to another one. And of course, the NASA postdoctoral program and the ENIGMA grant, and and Microsoft, who gave me some compute credits to run my big Looking Glass model on, which took more than 2 weeks and a pretty big machine. So that is always very helpful. And with that, I will stop talking and take some questions.
So we're a bit over time. So if anyone has a quick question to Adrienne, that would be okay. But
I guess if there's time, I have a quick question. Kara here. I think that's very, very interesting, and I have a lot of different things that I'd like to follow up with you and talk to you about. But I guess just one thing that stuck out to me in thinking about trying to interpret this wealth of work you've been doing is So when you talk about accuracy in your prediction and then going into the application of predicting, so I guess I'm curious about false positive rates. I guess that would be, say, more of a hindrance if you then go, if you have a rather high false positive rate in your model, and then you're going to predict in the ocean and finding 20% more sequences, and then want to do some downstream analysis of this, say phylogenetic or evolutionary analysis. I guess this could cause some issues. So one is, are you doing any control onto what kind of error you're having in your model? And then second, how do you go and deal with, going into a new data set and then sort of going to this next step of making sure you're certain what you're looking at is what you were hoping to find?
Yeah, so I think in terms of regulating false positives. Sorry? Wait.
I said, oh, I forgot that you had this one, this figure.
Yeah, yeah.
Oh.
So you can sort of toggle this probability threshold to cater to a particular precision level if you care about, really not having any false positives, or you want to, capture more of the potential oxidoreductases at the cost of precision. In terms of, there was another part of your question that I wanted to answer.
Yeah, so I guess it was in the predictions phase. Now you have 20 times more. Yeah.
Yes. So I did there. It's hard to validate with complete certainty, but I did run the MyPhaser annotation on these, and of the ones that they agree, one of the ones that were both annotated by MyPhaser and I had predictions for, there was very close agreement in the predictions themselves, and it tracked very well again with this precision curve. Like it was about 80% agreement. If you change the probability threshold to 0.9 or above 0.9, the precision went up to about these numbers again. Slightly worse, I think instead of 99, it was like 98 or 97 or something. But yeah, so that you can't be 100% certain that the MyPhaser predictions are correct, so it's hard to say exactly, but I think that's an indication that it's pretty good. And I actually did, I had tried training this model with a different way of splitting the data before, where I just split on the EC number, but not on the sequence identity. So, and that one I found a lot more overfitting, where when you went to the metagenome, the agreement between the MyPhaser predictions and the my own model predictions were much, much lower. So I think that does indicate that the one that I'm presenting here is a lot less overfit, and is pretty reliable.
So just for the sake of time, because I see Bob is ready to go, let's think.
Yeah, let me.
And wait, hopefully, Adrienne, you can talk to Kara offline.
Yeah, absolutely.
And, let's thank all the virtual applause for all the presenters. So thanks a lot. And I'm going to move on to theme 3.